Hi guys, this is Michael from Bridge Channel, and today, this video will talk about AP Bio Unit 1, Chapter 2, the Macromolecules. Let's jump in. In AP Bio, there are four big macromolecules. The four ones will be proteins, carbohydrates, nucleic acid, and lipid. And let's look at the subunits of these ones. For proteins, they are made of amino acid. For carbohydrates, they are made of monosaccharides. For nucleic acid, they are made of nucleotides, and for lipids, they are usually made of glycerol and fatty acid, but they are not always made of these, so you gotta be careful. In this video, we're gonna mainly focus on proteins and carbohydrates, and maybe if we have time, we're gonna talk about nucleic acids and lipids too. Now let's talk about proteins. Proteins usually make up the structure of cell and enzymes, and these proteins are made of carbon, hydrogen, oxygen, nitrogen, and sulfur. Proteins are made of polymers of amino acid, and they are usually three-dimensional structures. And this is how amino acid looks like. They are made of amino group, the central bone, and carboxyl group. And there are many types of amino acid in our world, and these kind of amino acids differ due to their R groups, which are bonded to the central carbon over here. And this R group determines the characteristic of these amino acids, and they can be rather hydrophobic, hydrophilic, or other special characteristics. And when this amino acid is ionized, they turn into something like this. This ionizing part is not that important, but well, it's better than not knowing it. So, when two amino acids bond together and form a protein, we call that a peptide bond. And peptide bond is a dehydration reaction, which means that H2O is created and it leaves the molecule. If you look at the picture over here, there are two amino acids, and when they come together, H2O goes away. And then the bond is formed. And we call this peptide bond, and this one over here is called polypeptide. Now let's talk about some characteristics of amino acid. So, amino acid sequence, which we call primary structure, determines how protein folds, which means that it determines how protein functions. And the directionality of protein, well, it begins with amino acid at the start and it ends with carboxyl usually. So when they form a bond, it's going to be something like this. Now let's talk about the secondary structure of protein. So protein, there are two kinds of secondary structure. The first one will be alpha helix and the second will be beta sheet. So the alpha helix means when the backbone of protein is twisted, which looks something like this. It's a bad drawing, but that's how it looks like. The beta sheet is when hydrogen bonds from two close chain of amino acids come together. And each sheets run in same or opposite directions. If they run in opposite direction, they're gonna look like this. And let's talk about the tertiary structure of protein. It just basically means that it does three-dimensional shape of peptides. That's all it says. And the quaternary structure is two or more polypeptide subunits interacting. And the biggest example will be, the most famous example will be hemoglobin in our blood, which transports oxygen. And this is a really important part that comes out in AP Bio, but except for primary structure, secondary, tertiary, and quaternary can be denatured. And this is because primary structures are simple. They're usually made up of like one line or, yeah, that's all that it's made of. But secondary, tertiary, and quaternary, they're really complex structures. And when we say it's denatured, these complex structures turn into simple structures such as primary structure or something more. I mean, they really don't talk about what they're going to become, but when they say denatured, only secondary, tertiary, and quaternary can be denatured. Now let's talk about carbohydrates. Carbohydrates are mainly made of carbon, hydrogen, and oxygen, and they are usually the source of energy, and they are composed of cell wall. They compose the cell wall of plants and fungi. And when we say source of energy, it's really easy to find it in our real life because, well, if when you want to study, you usually eat something to get energy for studying. And when you work out or when you go to school or when you try to do something active, then you usually need some source that gives you energy, which will be some foods such as rice, bread, cereals, or corns, or whatever. So, the subunits of carbohydrates will be monosaccharides, which are made of carbon, hydrogen, and oxygen, and they are usually in 1 to 2 to 1 ratio. This is not that important. This is usually, so it's not really important, but it's just good to know. And the subunits, which are monosaccharides, they are usually in linear or ring form. And this is the most famous one that we know, and its name is glucose. Usually we call it as 66 carbohydrates. We're going to talk about this later in Unit 4. Let's talk about the types of monosaccharides in AP Bio. The first one will be glucose, which I talked earlier, and these are the main energy source for cells, which are used in cellular respiration. And we usually call them as 6C, which is 6-carbon sugar. And for ribose, it's 5C, 5-carbon sugar, and it's used for RNA, and it stands for the first R in RNA. And deoxyribose, this is used for DNA, and this is also 5C because they are 5-carbon sugars. Let's talk about disaccharides. They are made of 2 monosaccharides, and, well, this 2 means the di in the front. And there are sucrose, fructose, lactose, and maltose. And the ones that you need to memorize are sucrose and fructose because sucrose are used for e-storage in fruits and honey, and fructose is used for sugar transported throughout plant bodies. Polysaccharides are the complex carbohydrates with more than 2 monosaccharides, and they usually refer to starch, glycogen, and cellulose. For starch, they are e-storage in plants, glycogen, they are energy storage in animals, and cellulose, they are structure material in plants. Well, I think we have some time, so let's go over to lipids. Oh, I thought nucleic acid were first, but anyways, let's go into lipids. So lipids are also made of carbon, hydrogen, and oxygen. They are usually hydrophobic, and which means that they are not soluble in water, and they are not polymer structure. The polymer structure that we learn in AP Bio are carbohydrates and protein and nucleic acids. Those are all. Let's talk about type of lipids. The major things are fats, oils, wax, steroids, phospholipids, and triglyceride. Wax are the waterproof on leaves, and they are usually made of fatty acid and alcohol. And steroids are the membrane of eukaryotic cell, and they are usually made of carbon atom group and other functional groups. Phospholipids, these are important. They compose the cell membrane, and these are made of phosphate group plus fatty acid plus glycerol, and they usually look something like this. And we're going to learn this in Unit 2. And the triglyceride is just the energy storage. Well, these are extra information that it's good to know. It's fine if you don't know. You just need to know that they're usually made of fatty acid and glycerol. Let's move on to saturated and unsaturated fat. Well, when you guys look at the nutrition facts behind like energy bars and stuff, they say that they have low saturated fat, they have no unsaturated fat or whatever. They really talk a lot about these. Well, we don't really learn specifically about their nutritional function in AP Bio, but we just need to know what they are. So let's talk about saturated fats. Saturated fats, they are called saturated because they don't have carbon-carbon double bond inside them. And unsaturated fat, they are called unsaturated because they have one or more carbon-carbon double bond inside them. This is all. This part about van der Waals force, it's good to know. Not really needed, but yeah, it's just good to know. Now let's talk about nucleic acids. Nucleic acids are kind of tricky but really important one in AP Bio because we're going to learn really deeply about this in Unit 6 and maybe a little bit of Unit 7. So nucleic acids, they are usually made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. And this is important because nucleic acid is the only macromolecule that uses phosphate for their composing themselves. So the subunits of nucleic acids are nucleotides, and these nucleotides are made of base plus sugar plus phosphate. So, and the sugar inside them, in DNA they are called deoxyribose, and for RNA they are called ribose. And it's really easy to memorize this part because DNA starts with D, which refers to the deoxyribose, and RNA starts with R, which refers to ribose. So this is the basic structure of nucleotides, which starts with phosphate group, ribose and deoxyribose, and the phosphate part. We're going to learn a little bit deeper about this later on, but yeah, you just need to know the basic structure of how nucleotide looks like. And when they bond together, they're going to form something like this. They're going to bond with something that looks symmetric with them, and well, this is going to keep going on. Let's talk about the nitrogen bases inside nucleic acids. There are 4 different types, which are called A, G, C, and T. And they all have their own name, which is adenine, guanine, cytosine, and thymine, but you're going to learn this later. The name is really not important, but you got to know that there are 4 big types in nitrogen bases. So for A, and G, they are usually called the purines, and they are formed as double ring structure. And different from A and G, C and T are called pyrimidines, and they are single ring structure. The one important part inside in here is that A, G, and C and T are for DNA. And when it turns into RNA, they are A, G, C, and U. When 2 nucleotides are bonded together, they are linked by phosphodiester bonds. You don't really have to know the name, but it's just good to know. And their directionality of nucleotides starts from phosphate group, which we call 5', and it ends as hydroxyl group, which we call as 3'. And these A, G, C, and T, they form a sequence, something like A, C, G, T, A, G, C, T, A, dot, 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 dot. And this sequence is really important because they contain the genetic information that we need for our lives. These are the ones that determine how we look, how our muscles grow, how tall we are, how healthy we are, and other genetic diseases and everything. So this is really important. So now let's talk about DNA. So DNA is a double helix structure, which means that it's made of something like this. It's made of something like this. And it's an anti-parallel direction, which means that this part will be 5 prime, 3 prime, and this will be 5 prime and 3 prime. This means that this one it goes from 3 prime to 5 prime, and this one it goes to 5 prime to 3 prime. That's what it means by anti-parallel structure. And when we talk about the double helix, A and T are the ones that respond together, and C and G are the ones that respond together. And when I say response, it means that when two nucleotides bond together, the nucleotide with A will bond with the nucleotide with T, and the nucleotide with C will bond with the nucleotide with G. But when they bond, A and T form 2 hydrogen bonds, and C and G form 3 hydrogen bonds. This one is really important. This might come out in AP Bio test because it's really small detail, but something that's really important. And for other type of nucleotides, there's something called ATP and cyclic AMP, but we're gonna learn this later in unit 3 or 4. Yep, that's all for today. Thank you for listening. And it was the note about macromolecules of Unit 1 in AP Bio. And if you want to take a closer look at the notes, I'm going to post it on my channel description, so it will be easy to access it. Well, thank you very much for listening, and see you next time.